All right, our final speaker of the day is Lauren Westhaver. Lauren Westhaver is a PhD candidate in the Department of Pathology at Dalhousie University under the supervision of Dr. Jeanette Boudreau. Her research focuses on the response of lymphocytes to endogenous signals of tissue damage and the regulation of sterile inflammation. Her presentation is entitled Quantitation of Mitochondrial Damage Associated Molecular Patterns in Perfusate and Bile for Analysis of Donor Liver Quality During Ex Vivo NMP. There will be a short final Q&A after the presentation. Thank you.
Hi there. I'm Lauren Westhaver. I'm a PhD candidate at Dalhousie. I'm also very fortunate to be a CDTRP trainee. So I'm kind of at the end of my graduate school journey, but kind of reflecting back, I've had so many opportunities and access to resources that I never would have had otherwise. So I'm very thankful. And I'm also very thankful to the organizers for allowing me to tell you about a kind of tiny offshoot of my main thesis work. So an unavoidable consequence of liver transplantation and really any intervention that's causing non-programmed or pathologic cell death is the release of intercellular components, which are collectively known as DAMPs. Normally, I have to wax poetic to get people to care about DAMPs, but thank you, you've done the legwork for me. So DAMPs is really just a kind of umbrella term for intercellular components that are normally involved in kind of homeostatic cell function. And then once the cell kind of releases them to the extracellular environment, they can be subject to immune recognition. So these are things like ATP, reactive oxygen species, DNA, and also mitochondria. And so what I study in my PhD is mitochondrial DAMPs, which are just DAMPs derived from mitochondria. And what we know, what others have shown in the literature, is that innate immune subsets like neutrophils are activated, become inflammatory in response to these mitochondrial DAMPs. And so what I spend a lot of my time thinking about is kind of a lymphocyte immunoregulation, conversely. So we also know that after an inflammatory response, the immune system needs to become immunoregulatory to return to baseline or to homeostasis. And so what we've actually shown is that lymphocytes, so later kind of responders to inflammation. This is a transplant crowd, so I'm showing you T cells, but the same is true for NK cells, will actually respond in an immunoregulatory manner. So here I'm showing you proliferation in T cells. This is CD3, CD28 driven proliferation. I'm showing you a dose-dependent decrease in proliferation in response to these DAMPs. We also see a decrease in inflammatory cytokine production in cytokine activated T cells. I'm showing you that here in the top right. And we've also shown in some in vivo models that the presence of DAMPs can interfere with the formation of antigen-specific T cell responses, both in primary and recall. So hopefully I've convinced you that DAMPs are really cool and they're important. Okay. But what I'm really trying to get at is that you have this kind of group of molecules that have really important immune consequences. They have a really profound effect on the immune system. And if you're an immunologist at heart like me, and you'd like to read more about this, we just published this in Cell Reports. Okay, but we also note about mitochondrial DAMPs, and in particular mitochondrial DNA, which are an important component of mitochondrial DAMPs, is that they serve as this robust and stable biomarker that's predictive of clinical outcomes. So, for instance, things like mitochondrial DNA in serum or cerebral spinal fluid, or even in urine, can predict things like ICU mortality, kidney injury, severity of ischemic stroke. And so given kind of the stability of these and the fact that we really wanted to be able to quantify mitochondrial DAMP release in some of our in vivo models, we decided to kind of work up a method to quantify these. So we first had to choose our targets. So we chose 4 mitochondrial genes, 4 because scientists love redundancy, but it could work with just one. So these are all coded for in the mitochondrial genome, and they're all members of the electron transport chain. And so what we chose to do is to order synthetic oligonucleotides. So these are of the target amplicon. So you basically, by serially diluting it, can have a standard curve that's specific to the primer pair that you're looking at. So basically you send them the sequence of your target amplicon, and then they send you back the double strand of DNA, and it's very, very cheap. And you have kind of batch to batch consistency in your standard curve. So if you're someone who likes looking at amplification curves, I am. My supervisor says it's weird, but I like the curves. So I'm showing you the amplification, the standard curve, and also some milk curve analysis. Here's what it looks like in E. I'm showing you on a gel. So it amplifies really cleanly. It's easy to work with. So our first question was whether or not our kind of devised method was as accurate as the kind of current gold standard, which is droplet digital PCR. So droplet digital PCR can do absolute quantitation, but it doesn't need a standard curve. Unfortunately, it's about 10 times as expensive as standard qPCR. And a lot of clinical labs don't have the kind of specialized equipment that's needed for droplet digital, but everyone and their dog basically has a qPCR machine. So what I'm showing you in yellow is kind of just a control. So it's whole mitochondrial DNA isolated from a liver cell line. And you can see that that's not as accurate as droplet digital or our synthetic oligonucleotide standard curve method. So we found that the values between our method and the kind of current gold standard were very similar. So they were non-significantly different. And so we kind of wanted to see if this was useful in a clinical setting. So we were very, very fortunate to get these samples from Dr. James Shapiro's group at the University of Alberta. So they had done a first in North America trial of normothermic ex-vivo liver perfusion prior to transplantation. So we were very fortunate to get perfusate samples from this study. So massive thanks to them. They were really great to work with. So these were livers that were kind of perfused and then transplanted. And so we asked first whether or not we could even quantify mitochondrial DAMPs within the perfusate and the bile collected during the study, and also whether or not this would correlate with some important clinical parameters. So here I'm just showing you correlation between our different mitogenes that we quantified, and as expected, they do correlate, and it's really not that exciting, but just kind of verification. And here's what the quantification actually looks like. So each kind of row is a different mitogene. Each kind of vertical column is a different donor liver that was perfused prior to transplantation. And then each dot is a progressive time point over the course of perfusion. So regardless of the mitogene, we kind of get similar answers. In about 7 of the livers, we saw a progressive increase in mitoDAMPs over the course of perfusion. And kind of our early analysis indicates that these might be associated with a greater incidence of early allograft dysfunction. So we're excited about that, but grain of salt because it's a very small n. So this is what I'm most excited about. We were actually able to isolate mitochondrial DNA, and it amplified beautifully in bile, which was surprising because it's bile. It's very caustic, but I'm showing you all 4 mitochondrial genes for the livers that did. So this is N of 18 produced bile while they were on the perfusion apparatus. So we anticipate that this may be a metric for biliary health, or kind of looking at tissue damage coming specifically from the biliary tree, which we're excited about as well. So then finally, we asked if this was correlated with any of the kind of clinical parameters that we had, and we saw significant correlation. So higher DAMPs indicating more cell death with warm ischemia time, unsurprisingly, cold ischemia time, donor lactate, donor INR, and we actually saw a negative correlation with IVC kind of outflow from the liver while in the perfusate apparatus. Okay, so why does this matter, and maybe why should you care? Well, mitochondrial DNA is a stable and robust biomarker. It can be applied to many different clinical scenarios. So we've done it in human serum and bile and perfusate. More recently, we're looking at levels of mitochondrial DNA in healthy patients versus patients with pancreatic cancer. And I think we're doing glioblastoma next. So this is my shameless plug. If you have any neat samples you would like quantified, I'd be more than happy to do that for you. And with that, I would like to thank, of course, the CDTRP, my supervisor, my committee members, in particular, Dr. Boris Gallo-Lopez, who secured these samples from Dr. Shapiro's group for us, my lab members, and all of you for your time and attention.
Very interesting. Thank you very much. Are there any questions from the online community? No, and anybody in person with questions? Here we go.
So first of all, thank you for putting CDTRP boldly on your beginning slide. It was well noticed. Well, I think the collaborations that led to this study, I think, were really very helpful to the community. Do you see this having importance in organs other than liver?
Well, I've actually seen some papers coming out about ex-vivo lung perfusion, and it's not limited to humans either. So kind of the reason we developed this method was I was looking at mouse models. So you swap out the primers for mitochondrial specific mouse primers, and you can do exactly the same system. So I was looking at it in a model of DSS. So it's not even limited to organ transplantation, any kind of model with tissue injury. So if you have bank samples stored away in your freezer that you centrifuged the cells out of and you'd like to quantify the mitochondrial DNA, it's a really simple technique, and I'd be happy to send anyone the primers as well.
It's a very fascinating talk.
Thanks.
My laboratory studies on mechanism of clearance of these necrotic and necroptotic cells before they release all these things. And so what's so unique about, when I think about DAMPs, I just think about all DAMPs. I was kind of naive till I came to your talk today. So thank you for illuminating me. What I want to know is what's so unique about mitochondrial DAMPs versus non-mitochondrial DAMPs? Is there some signatures on these proteins?
So it's a great question. And it's something I could definitely wax poetic about, but so the main difference with mitochondria with respect to liver transplantation, the liver is such a mitochondrial rich organ, but also there's, especially with respect to innate immune cells that have pattern recognition receptors, the mitochondria resemble, in terms of molecular patterns, their bacterial ancestors, right? So this will drive an inflammatory response. In my work, what I can really speak to is that we found an arginase dependent mechanism. So in other words, we had local depletion of arginine, which is really what's suppressing these T cells and NK cells in our model. So the liver, of course, has a unique isoform of arginase, but it's also found elsewhere in the body. So there's two isoforms. So there's many, many cool things about mitochondrial DAMPs. I'd love to talk to you about them, is the answer.
So I'm getting my mask stuck on my glasses here, per usual. It's a really great talk. Thank you for that. So can I ask a couple questions? Did you, for the mitochondrial DAMPs, did you notice, because you looked at, for instance, 4 different ones, did you notice any patterns that would indicate is one more helpful than another in terms of predicting liver damage or graft dysfunction, or any kind of patterns amongst relative expression? And the second is, because the organox is in essence a closed circuit, how do you tell about reaccumulation? So like if you had a DAMP filter, which I know some folks are using, could you, because if you start at a high level, you're thinking about like, does the organ get better, or does the injury, decrescendo?
Mm-hmm.
But once they're at a high level, you may stay at a high level. If you have a DAMP filter or something to remove the DAMPs and then look at rate of reaccumulation, would it potentially help determine usable liver from non-usable liver? So because I don't understand actually how they're cleared, to be honest. DAMPs are cleared in that context.
So the reason we really looked at this in the perfusate was because of a great question from my committee member, who kind of said we don't really have a great metric for perfusion. So things like AST, ALT, grafts that go on to be produce bile may have early allograft dysfunction. So this was kind of getting at that. I think the strength in this study is really just proof of concept. But we did see with a few livers that had, I can go back, progressive increase over the course of perfusion time. Those ones had, I think, a greater incidence of early allograft dysfunction, but really that was like an N of 7, and this is a single center trial. So it's hard to say for sure, but
Because they start high and go higher in essence for those ones, correct? Did they go down at all, or with a closed circuit, or you'd have to actually remove them?
You'd have to remove them because mitochondrial DNA is so stable, which makes it a great biomarker. But I mean, you can pull mitochondrial DNA out of early hominids in some cave in Patagonia, right? So they're there and they're probably going to stay. With respect to a DAMP filter, I don't know because they're small, right? They're small fragments of mitochondrial DNA. And then your first question was whether or not we found one was better than the other. So when we kind of looked at the correlation between our mitochondrial markers, which we didn't know, so we chose 4, we found that they all gave us the same answer. So kind of regardless of which one we looked at, they kind of gave us the same answer. Some correlated better than others. So I think if I had to choose 2 going forward, I'd probably do ND1 and ND6 just because the R squared value is like 0.99, but
Great, thank you.
Any more questions? Okay, wonderful. Thank you very much.